So, good afternoon, everyone. I think we can start now. So today we'll have a talk by Aliaksandr Halavatyi and also joined with Manuel, Gunkel and Marco Cosenza. And this talk is by the team from ALMF, which is Advanced Light Microscopy Facility at EMBL. And today Aliaksandr will present their work on adaptive feedback microscopy. And I will just quickly introduce Aliaksandr and the other people who participated in this work. So Aliaksandr studied physics, mathematics, and programming at Belarusian University. And then he did his PhD at the University of Luxembourg, working on the actin cytoskeleton dynamics, after which he joined EMBL for a postdoc, where he combined fluorescence microscopy and mathematical modeling to study kinetics of the essential secretion steps. And since 2016, he's working at ALMF here at EMBL, where he supports users for various microscopy techniques and automated microscopy workflows, which he will talk about today. And then Manuel Gunkel is a staff scientist also at ALMF, and he is providing here support and consultations in various microscopy techniques, including the topic of today's talk. And he did his PhD in physics at the University of Heidelberg in the single molecule localization microscopy field. And there he gathered experience in user support during his work in the advanced biological screening facility in the Bioquant Center at the University of Heidelberg. And finally, Marco Raffaele Cosenza is a researcher in cell and genome biology, and he has a deep expertise in imaging techniques and imaging analysis. And he did his PhD in a DKFZ, German Cancer Research Center, which was supported by the Helmholtz International Graduate School Scholarship. And there he applied advanced microscopy and imaging techniques to characterize a new source of cell division errors in cancer. And later he worked as a postdoc at EMBL, where he led a project to apply cutting-edge automated microscopy, machine learning, and single-cell technologies to study genomic variation in cancer. And since 2023, he continues to work on this line as a research scientist in the origin and function of genetic variation group at EMBL. And so I leave the floor to Alex, who will give the main talk and also probably will introduce the ALMF a bit more to the Janilia audience who might not be familiar with it. Go ahead.
All right, Nikita, thanks a lot for the kind introduction. Do you hear me well?
Yeah.
All right, thanks a lot for everyone for joining, and it's my pleasure to present the team with which I'm working and the work which we do in respect of the microscope automation. As Nikita already mentioned, I speak a lot on behalf of my colleague Manuel Gunkel and one of our power users, Marco Cosenza, who first started to use our workflows and then did extensive work to extend them and make them even nicer. So we will reach as well to that point. But maybe just to get into the topic and to set the scene, I would like to briefly introduce the team which I'm working on, the Advanced Light Microscopy Facility. So we are not a development unit, we are a service unit which we provide light microscopy services for the EMBL Heidelberg, but also for the external users. And these are the current team members. We are headed by Rainer Pepperkirk, and we are currently 5 people who provide the service. So we provide a lot of basic consultations, but on top of that, we have our specialties. So me and Manuel do a lot of different automation methods. Beata is specialized at the high throughput microscopy and assay development. Marco is our super resolution specialist, and Stefan also takes care of many different techniques, including SPEAM and also laser ablation methods. We also have the joint person between us and GSK, Giri, who develops this special omics services. So basically, what we do, we don't acquire the data for people, but instead we teach and support people how to use our microscopes, how to consult how to prepare the sample. But our major focus, of course, on image data acquisition and also on the analysis on and documentation on those images. We do a lot of development of simplified application protocols by, for example, by automation. So we set up, test, and offer cutting-edge equipment very often in collaboration with partners, and that's also an important aspect when we talk about automation on microscopy, and we organize different courses on light microscopy and also contribute to image analysis courses. So what is our equipment? So we don't, as we are as a facility who provides people to use the microscope, so we mainly don't work on the custom built microscope, but mainly we have like commercial microscopes from different vendors, and we cover a broad range of techniques as presented on the slides. So we have set of wide field microscopes, including TURF, but we have a lot of demand and a lot of focus on different optical sectioning techniques. There's laser scanning confocals. I mean, like a large number of different vendors with their own specialties. There's also like the spinning disc confocals and the light sheet microscopes. There are microscopes like wide field and confocal microscopes which are dedicated to more high throughput workflows, where we set up either screening protocols or advanced automation. And there's also important part is to the super resolution microscopes, where people want to push beyond the diffraction limit. The important part of our work is not only use the microscope as an imaging technique, but also use we have a lot of work on doing like different kinds of photo manipulation with the samples. For example, there's a lot of assays for our users which require photo bleaching, photo activation. We do have the special setups for the ablation or optogenetics experiments, and then we have also like systems for the laser micro dissections of the target objects. And now we come to the point why we want to actually automate all of that, right? Or not all of that, but in many cases, the automation is essential steps of internal process, because for the real biological study, which requires a lot of statistics or collection of different type of sample, very often the limiting factor is the throughput, right? We want to collect not one sample, but we can connect many images, or we want to isolate many cells for further analysis. We need to target the right object or object state. For example, if we want to talk about high resolution, right? We cannot image everything at a high resolution in a short time, but we want to use an automated protocol to find the objects and actually zoom into them and to acquire them. And as I mentioned already, there are different high content modalities which are involved, and we need to be able to fast trigger on the fast respond of the sample changes to trigger those modalities. And also very often we need to correlate images acquired at different modalities afterwards. So this is the classical example, right? As just one would provide as a textbook. So if one interested in mitotic cells on the confocal microscope, so the regular user who will sit in front of the microscope need to acquire a low zoom image, find these cells, right, and basically mark these positions and trigger the corresponding high resolution acquisition, or maybe in 3D stack or 3D stack, multicolor, and so on and so forth. This requires considerable amount of time. First of all, this limits the throughput, and second, by the time the user does all of that, the sample might already change and the right states are not captured. So now we come to the basis, what we call the definition of the feedback microscopy workflow, but many people, of course, will use other terms. It's terms like smart microscopy, intelligent imaging, and so on and so forth. One can find to the literature. So that's basically, again, the basic textbook loop, which many of you know. So we typically would have the multi-position experiment where the microscope will be configured to completely automatically perform such kind of a loop. After moving to the next position, it will perform the autofocus and the image, like low zoom resolution image to be acquired. And instead of user providing the feedback, this to be sent to the image analysis routine, which can be inbuilt in microscope software, or it can be open source, like ImageL, Cell Profiler, etc. And I will come back to the different options later. And then from that, when this object is automatically identified without any user interference, the coordinate of this object is basically sent for the high content imaging. It can be 3D multicolor imaging, or it can be FRAP, FCS, or any modality which we want to think. After all the identified objects in this position are imaged or corresponding experiments are performed, the microscope moves to this next position, and then the loop performs over and over again. So that's basically where it all started, and the facility has long-term experience with doing and setting up such kind of workflows on different commercial microscopes. So before we go to the technical implementations, let's just have a look to the few more examples to see the variability of different assays which we need to deal with. So again, another example with the targeting imaging, where we have like a multi-well plate with Drosophila embryos, and each of these embryos or embryo with a specific phenotype needs to be imaged at high resolution. Of course, imaging its brutal force will require like infinite amount of time and will generate infinite amount of data. Instead, we perform the same logic. So we identify all the embryos with by scanning with a low magnification objective, and we then switch for the high magnification objective to the for the confocal imaging. As I said, beyond imaging, we do a lot of F techniques, and that's actually the example of which comes from my postdoctoral project. So here we needed to perform FRAP on small structures which are called ER exit sites, and the issue is that this structure, first the recoveries are very fast, so one has to basically be very fast to configuring this experiment, and then they're also mobile inside the cell. So basically the experiment, which one can also perform manually, looks like that. So we need to zoom in into a certain part of a cell, right, where consists of structures of interest. And then we select basically the region which needs to be photo bleached. And after this region is selected, so the photo bleaching can be triggered, and we can actually observe the recovery over time. When this recovery is calculated, we can basically segment and track the structure, and basically by quantifying the fluorescence intensity over time, we can conclude actually about mobile and immobile fractions, and also by analyzing the rate of the recovery, we can conclude on how fast binding and binding kinetics on the structures happens. Of course, there is a possibility to perform this experiments manually, but in real cases, it's a very tedious task, first of all, because one has to always switch between low zoom and high zoom modalities, and then select and draw these regions of interest. And maybe by the time you draw this in the region of interest, and the structure is not anymore in the area of interest, right? So one has to basically start experiment over and over again. So manually is kind of very low success rate. So another example where the automation of photomanipulation experiments is essential is use of photoactivation for phenotypic targeting of the cells. So here we use also basically the photoactivation effect, where we use the UV laser to basically to mark certain cells, but not for the purpose of studying kinetics, but for the purpose of marking specific cell subpopulation. So briefly, in this project, people were interested in the mixed cell population. Some of them were having compact Golgi phenotypes, which is depicted in green, and some other cells having like fragmented Golgi phenotypes, to separate one subpopulation of cells from another subpopulation of cells to do a follow-up molecular studies of that. Of course, there are different methods exist, but one of the possibilities is to put the photoactivatable or photoconvertible dye in such a cells and actually mark to be able to photo activate the cells in one well with, for example, fragmented Golgi phenotype, in another well with compact Golgi phenotype, to be able to use the FACS sorting to isolate, basically mark subpopulation of cells from other cells, and actually go with any kind of molecule for example, sequencing studies is that. So it can be like single cell sequencing, or it can be bulk sequencing. And in all these cases, it's also essential to make it fast and automated for several reasons not to be on only not only for the high throughput. So normally you work in this case with a live cell, so it has to be fast, basically high throughput, like a lot of cells photoactivated in a limited number of time and as unbiased as possible. So it's also makes sense to give this task of finding the right cells to the microscopes instead of users sitting there and picking up the cells to be photoactivated. So I just highlighted a few examples, but they will be more later during the talk. Altogether, we have variety of applications, right? And here I have like a summary of different kinds of applications, but there are sometimes combinations of those. We were talking about special imaging, like special or rare phenotypes. Another application is tracing moving objects, where we have, for example, a crawling cell, and for the high resolution imaging, we want to move the field of view of the microscope by along with this moving cell. So this will be obviously on the fly, requiring the image analysis. There are tasks which require the correlative imaging, where we do the same as in the first case, but also switch the samples between different microscopes. And as I mentioned already, we do a lot of photo bleaching, photoactivation, and other photomanipulation studies in different flavors, which requires additional criteria for the microscope automation, which I will talk later. As adjacent task to that are the complex experiments, which basically require sample treatment on the stage, right? So in this case, we speak not only about different imaging modalities, but also manipulating the samples by, for example, connecting to the microfluidic devices, pipetting robots, or any other external devices for the sample manipulation, which might be needed to perform, for example, multiplex imaging experiments. So I mention it here briefly because normally we require the same type of the instrumentation, where we can actually trigger or synchronize different imaging modalities with the processes happening outside of the acquisition software. So being it image analysis, or being it's like external manipulation of the sample. And to mention, for the demanding applications, we start actually to combine very often, for example, the multiplexing by handling the fluidics with automated photo activation or cross-linking. There's such protocol required by them. So I will focus during the rest of the talk on the left-hand side, right? So with the examples which I provided in the beginning. So the part on the right-hand side is out of the scope of the talk of today, but I'm happy also discuss the different implementations and different protocols which we can get there. So now, after briefly introducing different tasks which we want to accomplish with image analysis, so let's basically talk about how we would do it in practice. Before defining the technical implementations, let's just consider what would be the typical workflow of a user which will use such a protocol, and what will be the criteria of the protocol being efficient and robust and good enough to perform the biological study. So typically, the workflow not only consists of the perform experiments, right, but we need basically the tools where we can design and debug workflows, right, the imaging and also the image analysis. And we have to obviously perform experiments. These experiments running in automated fashion will give us a lot of the data, and that will be the multi-scale data, low resolution images, high resolution images, right? So also data generated by different F techniques and so on and so forth. So one should be able to make sense of the data, maybe to browse the data, also browse the data in the efficient manner, and have all information about from experiment to be able to analyze the data in the high throughput manner. So when the user comes to me in the facility, right, I will basically think of a certain criteria which I need to think for the workflow to happen. So we need, so first of all, we need fast image analysis and communication between image analysis and the microscope software. How fast it has to be, of course, will depend on the application. So in some cases, few seconds, it's fine. In some other cases, image analysis of few seconds will be too slow to catch the phenotypes of interest. Very often, one feedback step, for example, jumping from low zoom image to high zoom image is not sufficient. So to make the workflow really efficient, we need multiple feedback steps. For example, we have like low resolution image, but then we have medium resolution image when we focus on the cell, and then we further focus on the basically on the part of the cell we want to image. We realize that by running a lot of workflows, they have, of course, always a differences, but they have a similarity. So it's important to be able to expand and modify workflows rather than define them from scratch, and have some kind of common tools which allow basically to expand the workflows. It's also important to log experimental and analysis info. So basically to have all the information about events which happen during this experiment, because we don't want only to have the images at the end, but we want, for example, to know what was going right or what was going wrong during the analysis, or we want to robustly find the high resolution image on top of the low resolution image, right? And make sense of all sense of all information and all the data which are produced during the experiment for the analysis. And finally, we want to have the workflows reusable, so they can be reusable, for example, for different object phenotype, or we also thinking how we can make it reusable in a sense that we can move workflow from the confocal microscope A and confocal microscope to the confocal microscope B, maybe of different brand, and what is required in terms of the software to make these protocols transferable. So in terms of the software implementation, there are two different scenarios which we can follow. And now I talk about the scenarios which we have on the commercial microscope, because I know that many of you build your own microscopes, and of course you can implement such workflows very efficiently by building your own software and make it also very flexible. As I mentioned already, we work with the commercial microscopes, and basically we try to see how far we can go with such workflow automations with the commercial platforms. One possibility is to use the inbuilt image analysis and decision making, which companies provide as additional modules. So they actually a growing market of such additional modules for different software vendors. The advantage for users is that the graphical user interface normally will be provided, and it's easy to design the simple workflows. The disadvantage will be the lack of, sometimes there is a lack of flexibility to image analysis, because if we use a company internal solution, we are limited to the methods which are implemented in this particular microscope software. They are doing it better and better, but you are not free to choose which maybe method you are used to have otherwise. These workflows might be limited for complex and multi-step workflows, because one often cannot freely decide all the logic on the workflow. So, and we know basically all the pros and cons of relying on the commercial software, in particular when it comes to the bug issues, right, or something doesn't work, and then basically how this needs to be fixed. The alternative way, which I will talk about for the rest of the talk, is basically to setting up the communication between the software. So what we want to do here, we want to have the commercial software to take care of the acquisition process and define the acquisition settings, but we want to actually combine the commercial microscope software with open source tools for image analysis to be able to basically to trigger the image analysis methods what we want, and also be more flexible in the control of the workflow. This gives us more flexibility and more control on what we actually want to do, but very often it's coding will be required for such an approach, and then it's basically would stop end user to implement such thing from scratch. So what our goal is here to provide the set of the tools and the protocol which will simplify the work for the end user, so that we basically that particular person coming with a particular problem needs like little to no coding to implement the task which they want. So how would it work? The general scheme of such a workflow will work like this. So we have a microscope. As I said, we work with the commercial microscope platforms of different vendors, and we have a microscope software, right, which will control the acquisition process. So normally the user will be somewhere here and will click the buttons in the microscope software and tell the microscope what to do. As we don't have the user now, we won't have the microscope software automatically say produce and save the images, and these images will be sent to the image analysis module, which can catch these images and then analyzing them on the fly and send the feedback to the microscope via additional layer, which we call here command interface, basically, which will translate the result of image analysis to command of them to the microscope software of what the microscope has to do next. And of course, the obvious requirement here is that the microscope software has to support such an interface. So with about the different types of the interface, I will talk later. So the first challenge which we tried to tackle here, assuming that we have such a structure, assuming we have a microscope and assuming we have a microscope command interface, because there's several brands which support such architecture, how to streamline the image analysis part and the decision making in order to make these workflows more configurable and flexible for the users. And that's what I started to do a couple of years ago, starting with my postdoctoral project, and then also by supporting users in the facility. I came over years, we came up with a feature library, which is called ALMF tool. So basically, it's a set of plugins and classes, which basically allows us to make the custom workflows on our choice. And that's how it will work. So basically, the part which is outside of the big box will happen outside of the ALMF tools, and inside what we have inside the box is basically the set of classes which will communicate, a set of classes or objects which will communicate inside of ALMF tools to basically make the analysis happening. So as I mentioned already, that we have a Microsoft software which we will save a new image file. And this new image file will come to the first layer, which will be called job distributor. So each particular workflow will have its own job distributor, which has nothing to do with particular analysis task, which will actually capture this image and define what has to be done with this image, right? So more often than not, that each image which is captured has a specific name tag, right, which identifies what is the job for the image analysis. And the job distributor has nothing else to do than actually to send this image to the particular analysis task, being its autofocus, being its cell identification, being its bleaching region definition, and so on and so forth. There are inbuilt jobs which we already implemented. There's also possibility to subclass these jobs, being like by subclassing the basic job class in Java or in one of the scripting languages. Another possibility which we provide and we implement, we have special jobs which don't do basically the task inside feature, but if people want to provide the custom elastic classifier or custom Python script, right, which does the job, then the image basically sent for the analysis, for more complicated analysis out of feature, and then the result basically will be translated back to the corresponding job. So after the job is finished, so it communicates back to the job distributor and sent actually via remote control interface commands to the microscope software what to do. On top of doing the job, as I said, we realized for debugging and also for running post-acquisition analysis, it's important to capture the information about all events happened during the acquisition, and unexpected, because in practice, if microscope crashes middle of the night, and I don't know whether it's image analysis problem or a microscope problem, I want to catch all essential information. For that, all the components of this workflow communicates to so-called experiment database table. So in practice, it's like quite simply structured temp-delimited text file, which captures information about all images acquired, but also about like all the decision made, for example, positions identified, ablations regions identified, and so on and so forth. And then we basically connect it to a simple visualization tool, which allows us to automatically to scroll through this table and see actual image analysis results. So that's about the structure now, and that's maybe a lot of the theory, and one would think why such a complexity is needed, so why you go through all of that. So I come now back to the examples which I was talking in the beginning and basically showing what we achieved with that. So again, the example where we're imaging like Drosophilus at high acquisition, So if one takes it by the textbook, one thinks that basically jumping from a low resolution, high resolution imaging is just enough of what we need to do. In practice, when we started to implement this workflow, the final workflow for us looked like that. So in reality, we had, instead of 2 image acquisition tasks, 4 image acquisition tasks, and instead of 1 image analysis step, 3 image analysis steps, to make this workflow happen on the hardware which we had at that time. So first, first 2 jobs are acquired with 5x objectives, and the last 2 are acquired with 20x objectives. First, when the microscope goes to a new well, so provided for the specific sample, the hardware autofocus might not work, or implemented commercial software autofocus might not work so well. So we have the EXI scan. That's actually quite simple image analysis where we identify a reflection of the cover slip. Then we acquire overview of the well, and it's only 1 embryo highlighted. That's each embryo. All required embryos will be segmented. And then before we jump to the final job, we actually switch to the 20x objective and make the sparse Z-stack in 1 channel. So for 2 purposes. So first, it allows us to center embryo in XYZ, right? Because they can be different orientations, and also like because of the sample mounting, be at a different height. And then because we can fit it to ellipsoid and can define the rotation. So we were able, for the confocal scanning, we were able to crop scanning area to the corresponding rectangle and save significant amount of scanning time without need to scan the extra area by jumping to that. So another example, I already mentioned the automated FRAP, and I said it's quite tedious to do it manually. Again, to make it really automated, we had to establish it as a multi-step workflow with the several decisions which need being made. So on here in this scheme, the top row represents what happens on the microscope side, and basically the bottom row represents what happens on the analysis side. Again, we start with a cover slip reflection. So that step, which actually very simple, but which we reuse over and over again. So it's just 1 job, and it's always defined, and people can use it for the different workflows. Then next step for the microscope to acquire the overview image, and which will lead to the segmentation of a target cell. Then and also the area which will be used for the FRAP. Then microscope will zoom in this area, and we have like a fast Z-stack already in the limited field of view to find the structure of interest and perfectly like focus on that in the best possible way. And then after getting the 3rd result, we actually start the FRAP experiment, knowing that basically the structure, we segment the right structure, and it's even like in focus. So it basically improves for us drastically the success rate of such starts. As I mentioned already, it's like normally during the 1-day session, if you're successful, you will collect maybe 20, 30 of these recoveries. Like in overnight runs, we were collecting hundreds of these recoveries. And this was allowing us not to basically get the average behavior, but also get the distribution of, for example, recovery times, and also basically have a look on the heterogeneity of such behaviors, right? Which one would otherwise would be very complicated to do with manually acquired data. So this is about bleaching. So what if we want to do something else? So we can also isolate the samples in different ways. So for example, this is the Leica microdissection experiment here at EMBL Imaging Center. And this is the project done by my colleague Manuel. So again, this is a commercial microscope, and in principle, there is software which allows to draw the regions and to microdissect different areas, which will be collected then in the caps of Eppendorf tubes. One can do it manually, but the throughput is limited. So we saw whether we can use our Automic tools logic, right, and basically our schema to be able to connect to the different microscopes by keeping the entire logic the same. So that's how it looked at the end. First, different imaging conditions and the imaging conditions and microdissection parameters have to be set up by the microscope software. Then in this simple case, right, we have like a user-defined macro. So this is just example of the test case, which can segment the target cells and have like a logic to prioritize which has ones have to be the which has to be isolated, and then one can define the multi-position acquisition, and basically at the end the microscope will on each next image run such a macro, right, find the cells to microdissect and collect them. Again, if the image analysis is already established for something else, for example, FRAP, we can simply reuse it and just connect to the different microscope by the layer which defines how the commands have to be translated to the particular microscope implementation. So another step about reusability, as I said, that was the initial project where the different phenotypes had to be photoactivated for the subsequent sorting. That basically triggered a lot of interest, and then we have the Marco Cosenza actually followed on that. So instead of analyzing golden phenotypes, then he is interested in basically photoactivating and sorting micronucleated cells. So if we follow the same schema and if we stay on the same microscope, the only module which we need to change basically there is the job which is responsible for the particular image analysis, right? But the rest about the autofocus and about the running the entire experiment logic can stay the same. We have now also tested how we can propagate such a workflows from the platform A, being like size LSM 900 microscope, which has API level, which allows us to communicate to Olympus Lorg 3000 microscope by keeping image analysis exactly the same and only translating the commands to the different microscope software. So the future work, which is now done by collaboration of different groups. So there's a big EMBL initiative, which you probably heard about. There's a track expedition which goes across the coast and collects marine samples and wants to do molecular biologists with that. So there's Tina Wiegand, who is basically mobile services support. There's a size LSM 900 microscope travels together with the expedition, and the idea is to apply the same this protocol with obviously more complicated image analysis, which will identify the target objects and basically to do the same type of a photoactivation assay to be able to sort and do molecular analysis afterwards. So this is a biological project of Thomas, and it's nicely supported by Chris Rhodes for the technical implementation and expanding our workflows further. So I hope I convinced you for the power and the need and basically the opportunities that which bring our modular approach to automate different kind of microscopy experiments on the commercial platforms. In the last few moments, I want just to highlight a few challenges which we tackle now and our ways to go about it, both inside EMBL and by making the collaborations across institutions, and also will be happy to hear your feedback on that. The first challenge, which I already mentioned, is actually the need of the command interface, right? So that's basically one can always have captured newly acquired images by monitoring the folder, but if we don't have the way to provide commands back to the microscope software, it will not do the job. Unfortunately, there is a big discrepancy between different vendors. Some support it as just some still don't support it at all. Others support it with very limited functionality that we can send some commands and cannot send other commands. We try to actually stimulate knowing the importance of such a workflows, and it's not only the feedback from our institute. We have the similar feedback from our facility. We try to basically to establish the smart microscopy working group where several facilities across Europe, but also academia partners contribute, where we want to have actually, which has several goals. So we want to have the standardized nomenclature of the feedback microscopy workflows to see what is the common ground, right? What kind of the modules and what kind of workflows would need to be established, and also suggest API specifications for different applications. And the goal is to come to the industry partners to say, okay, so if you want to be compatible with such a workflows, we suggest such a standard, and maybe by implementing such a standard in your software or in the different softwares across different vendors, we can just find a common ground and to provide a more robust flexibility to that it becomes easy in future to work with that. The next complication is actually how the workflow is controlled on the microscope side. Again, this is a little bit more detailed schema what will happen on the acquisition side. If we have a microscope software, we do a lot currently with size and blues, and we need a layer, right, which actually controls what needs to be done by the microscope. Again, it can be implemented on different microscopes. In case of size and blue, we have like a scripting environment, which is then macro, and where we can actually run the workflows, run another script, which will define the initial positions and which will control, for example, such a loop where it will start the experiment, try and define the position loop, and then run one job after another. If needed, wait for the feedback and come back to the next acquisition process, and so on and so forth. When we started with this plot from several years ago, again, each workflow on top of we needed like Atomic tools workflow on the analysis side, we had to make each time the new script for particular workflow on the microscope acquisition side. Of course, there's a lot of code application. So what is now started by Marco Cosenza, he tries to come to the common nomenclature, right? That we have basically the basic acquisition job step, and this is parameterized, which we can basically replicate, and we don't need to override the scripts over and over again. That we have one standard script and one standard protocol definition, which will work for different type of the workflows, because in fact a lot of decision making is happening now on the image analysis side, and here we can go further to have one standard application which will link this image analysis to the microscope software for the true complexity of the experiment which we want to do. Finally, the last challenge which we need to work further on is basically how we do about extendability of our workflows in terms of the image analysis side. As I mentioned already, each of this module of the modules in Atomic tools is a Java class, and it's always possible to subclass it to introduce a new functionality on the level of job distributor or on the level of particular job. It's also possible to provide the custom macro, the script, which gives which has current image is always an input, right? And the set of positions and the rows, which in the defined format is always the output. But we also want to bridge to other softwares, as we already mentioned. So currently it works either via direct system calls, for example, for Python, but it can be also for Elastic. We use nice Elastic for IJ plugin, right? Which the call to this plugin will be the part of a job. But we want what we realize that in other two cases, depending which kind of environment we run, there's a lot of overhead. For example, in case of Elastic, but also in case of Python, for repetitive image analysis, if you just do it via direct calls, basically environment has to be initialized over and over again, and it might significantly delay the analysis time. What Mark Cosenza also started to do, so he established like an alternative way to communicate, where he starts a fast API server, and the server can be run locally on the acquisition PC or on the EMBL cluster in our case. And instead of triggering the outside process each time over and over again, the server is waiting for images and actually analyzes them in much faster way, and then sends a feedback for the follow-up analysis. by increasing the robustness and the throughput. So this was done by Marco in respect mainly of his particular biological project. Now we actually try to see also together with Manuel and also Christopher Rhodes how we can also use the same type of scenario for other biological applications and also bridging other tools which require, for example, Python. So with that, I come to the summary of my talk. As I mentioned already, we were building a lot of the workflows on the Ultimate Tools library, which provides us quite flexible and extendable toolbox for designing simple and to advanced feedback microscopy workflows. We have compatibility with multiple commercial microscope platforms, and we have the solution to log and interactive browsing experiment workflow information. So that's our current and future challenges and developments. So we want to have better implementation for the machine learning routines which happen outside Fiji to make it faster and to also be able to include more sophisticated analysis on the image analysis side for the demanding applications. And the final goal in terms of the structuring of the workflow, which we foresee and hope will come in future, will hopefully look like that we don't have the workflow management on the microscope side and second workflow management on image analysis side, that we have sort of the central workflow manager, which will define the entire logic of the experiment, and the external modules will work as will just wait for the commands and give the feedback. And microscope, image analysis, and maybe microfluidic device or other devices which needs to be included will be like part of these external modules. And hopefully it will be easier to extrapolate and also to basically shift the same workflows between different microscopes, image analysis modules, and so on and so forth. With that, I would like to thank all people who contributed. I would like to thank all my colleagues from ALMF, especially Manuel. I would like to also mention Lukas Schutz, Volker Hilsenstein, and Sabine Reiter. So, as I said, that ALMF has long history by developing the infrastructure and workflows for the feedback microscopy. So Lukas, Volker, and Sabine already in different places by now, but we collaborate a lot with Center by Image Analysis, Christian Tischer and Arif Khan, because a lot of image analysis for the feedback microscopy is actually done by them, and then we incorporate it into the workflows. I mentioned already the contributors from the Peppercock team, so special thanks to Marcus. I already mentioned he's our user and at the same time contributes a lot by developing the workflows further. So a mobile services team is now just recently established, and we optimize the workflows for their demanding applications, and we have a lot of exchange and collaboration with Antonio Politi in particular for running experiments on size sand black systems where he develops the corresponding microsys. We thank to all industrial collaborators for providing the support and also for the feedback on our request, and also like open source image analysis community. So special thanks also to Elastic team here at EMBL for the support. So with that, I hope I'm not too much over time. So thanks a lot for your attention, and I'm happy to get questions.
Nice. Thank you, Aliaksandr. It was a great talk. So I see Mark can go ahead with the question.
Sure. Yeah, Aliaksandr, thank you for the great talk. It's very informative to see kind of what you guys are thinking of building out there at ALMF. I think my question was kind of looking at your workflows, and I see you're exporting the images to TIFF. I was also wondering what you're doing with the metadata and how you were figuring out how to standardize that across different vendors and microscopes.
Okay, so thanks for the question. Actually, the data handling is one of the big things in respect of the fairness, etc. So actually, the TIFF was mentioned here because that's how Marco implemented it to connect to the server. In our, let's say, the basic scenario, we work with the default vendor file formats. So the idea which I initially had, and we pursue in most of the cases, we don't want to translate the data on the fly to any format. So if the microscope basically, if the size microscope says the CZI files are captured here, right? And we analyze them. Actually, here we also document the information about the CZI files. We only do the processing which is essential to generate the feedback, but we don't try to convert to anything like OMETSAR or any kind of a standardized file format. So at this step, we preserve all the metadata which inside in the raw file formats, right? What should happen next? It's an open question, right? So there's a lot of the discussion. Of course, maybe they're keeping such a table which basically knows the process to all the files, not the robust way, but that's not the intention of running it at the microscope. As soon as the data are collected, one has to decide, or maybe we have to think more what should be the guidelines, how to translate or resave the data. But during the acquisition, we save data in raw file formats.
Got it. Yeah, I was just thinking because like some vendors like Slice do have some export capability to say only TIFF, right? And that at least give you somewhat standardized XML, although in my experience, these are all very different and all have their different quirks. So but it seems they're still trying to figure that out as much as we are. So
yeah,
it'd be great to have a solution for this eventually.
I mean, that should be, I mean, that's kind of the part of this working group, which I already mentioned, right? Just to see what kind of the information and how the data have to be transferred. But I think it's even the broader discussion which goes beyond the feedback microscope with OMETSAR initiative and trying to get to the standardized file format, right? But that's kind of indefinite story, right? Because that was the origin of to come up with the OMETI at some point. So we don't have a general solution with that, right? So we, for each of, actually what we do on the background, most of the time we use bioformats to open the native file formats. I mean, I have to name it. Yeah, so we benefit by the bioformats, and that's why we don't need to change the autofocus job, being it getting it CZI file or getting it OIR file from Olympus microscope. So that's basically very simple solution for now.
Right. And I think I know this task is also very difficult even for custom-built microscopes that we're building internally at Junelia, because I think it's hard to say that there's an established standard to do so at the moment. And even OMESAR just presents its own challenges in terms of as an acquisition of file format. And I wonder if you had any opinions about the state of OMESAR for your needs. And then I'll turn it over to Akita.
Well, as I just mentioned, so in our scenario, we work with a commercial microscope. So we are all for unified file formats, which will make it easier to making our data fair, right? Because there's a lot of discussion going on what we should do ourselves, right? Should we resave or should we not resave? If we resave, should we keep the original data as CZI or IR files, or should we not have, right? Whether we or we put them to the archive and do the processing on the OMETSAR data. And this is, again, not in the context of the feedback microscopy, but more in general of the fair data handling. So I believe that these are the discussions probably also going on regularly in the format of these meetings, but also in the other meetings. But there's a work, right, and progress on that, but there's no general solution. So for the feedback microscopy, original complexity is that you have this multi-scale data, and you not only have imaging data, sometimes you have these FCS curves, right, or you have this FRAP recoveries, right? So it's basically like not only multidimensional data, but sort of a different. So for now, I don't see like a generic solution how it will be packed in a file format. So hopefully OMETSAR gets more and more mature and we hear the solution about it. But if you have more suggestions or highlights how to do that, would be happy to hear.
I have a more political question about all those brands of microscope. Are they okay giving you access to their hardware, I guess? And whether you see those big companies like Zeiss building those products that you're working on now in the academic environment for industry? Like,
Yeah, that's an interesting question. Basically, maybe just to have a little bit of a history. So this work group, which I mentioned right on one of the slides, just maybe can just roll back to that. So the link is here. So it started by the one-day workshop, which we organized here at EMBL October last year, which we had like a company representatives sitting next to us, not only to our facility, but also to other facilities which were invited. So it was organized by the Europe Imaging Industry Board. And basically we were for an entire day discussing exactly that. So what basically are there, what is good and what is bad, they're providing the access to that. And that's, of course, a lot of the tension from the big company side. If we give you unlimited access, then what will happen if you run something crazy, right? Who will be responsible for that? The idea which we came at the end of this workshop, and that's the idea which we try to follow, basically this small working group is actually spin-off or was originated from this workshop. We said, okay, we don't want to modify your software. We don't want to communicate directly to the hardware. So actually, we intentionally want to communicate via your software because we want to respect all your safety layers, right? So we need at the end, for the workflows which I mentioned, we need at the end probably the given set of commands, right? And I mean, you can list them, right? Move to the next position, activate setting which is saved under a certain name, define the bleaching region, and so on and so forth. So the logic is that people should use the commercial software to define the imaging settings, right? And many things which can be predefined should be predefined in commercial software. Then more often than not, you have the logic where you have given a certain name for the acquisition configuration, right? And your script will trigger these different configurations at different positions. And for many workflows, it's simply what people need in reality. But already to convince companies to come to that on a common ground, right? It's already very difficult. Sometimes actually the smaller companies respond even faster than the bigger companies because they don't want to handle all this complexity of the workflows. They said, just fine, we deal with our acquisition side and we can open certain functionalities for you. The big companies try to sell everything in box, right? So there's all these image analysis modules in. So then it becomes a little bit more challenging. I think they also see now additional benefits by us developing more complex workflows, basically by communicating between the softwares. And then if they see the profit from such a workflow, right, that they implement it inside the microscope software. So I think,
yeah,
I think the future should be a combination of both. Because
yeah, that was my second question of fear, whether those bigger companies could kind of eat your solutions and incorporate into their new module for thousands of euros.
Well, I don't know whether it's our solution or solution by someone else, but for example, I mean, now just make an example right now. Zeiss for a long time didn't have the inbuilt solution for the photoactivation. They had the resolution for low zoom and high zoom imaging. Now they have also the photo bleaching photoactivation solution, right? Which basically with the inbuilt tools for image analysis can also bleach and photoactivate, right? And we cannot force them to do something, right? We try to convince them that it's also beneficial on both sides to have some kind of open layer, right? Which we can send the certain time of commands. But again, so when in the commercial setup and in the setup of the facility, I don't want to have a command to access the confocal scanner, right? Or I don't want to have even a command, maybe not even to change the laser power, right? So the idea is operate by the higher level commands, right? And being able to build the workflows from those.
Was there?
Hi, this is Damien from Janelia. I would have a question. So a very nice presentation, and I'm very convinced of the importance of developing this kind of approaches. And my question is, so I see here the advantage of this all custom solution, but there are also some commercial solutions like the jobs with Nikon or the high content screening automation with Leica. And I wanted to know if you could comment on what are the main limitations of this, like basically commercial or included that kind of software. Yeah.
So thanks for the question. So I mean, we are of course aware of all of these different solutions, and the limitations will be different in different cases. The major so if we want to find the common ground, the major limitation for me would be to have the access on the modality for a certain modality, which probably available for me on a certain microscope brand, right? But doesn't have basically the extensive workflow control on that, because very often we have them basically something which has the extensive inbuilt solution for the workflow configuration, right? And for the image analysis side. But the imaging modality or bleaching modality, right, is not available on this platform, right? Or you have other way around. I have very nice tool which can bleach or photoactivate, right? But I don't have the possibility of a workflow control. So ideally, I want to have both, right? But very often I have either one side of the story or another side of the story. Even inside the major brands, right? There are some products, for example, like the microdissection systems for Zeiss and Leica, they are not on the mainstream of their commercial software products, right? Because it's like a very special product for that. Sometimes they develop very special software with a reduced number of features. Another limitation, but not the limitation, but the factor we have to consider, right? It's about how many licenses and how many different applications we can maintain in the facility workflow. Because if I want today do feedback microscopy on Zeiss experiment, tomorrow on Leica, afterwards on Olympus, let's assume that they have all the inbuilt image analysis modules. First, I need to know all these image analysis modules, and I need to be able to train the users on those, right? Because it's basically maintaining even more solutions on that. And second, it's also about the licensing issue, right? So then you need to have all these image analysis modules and also hopefully up to date them to be able to run all of that. So we don't discriminate the inbuilt solutions. Actually, we use also them in the facility, right? But in real world scenario where you want to have modality X, which is implemented just on a particular brand, but maybe either not available or less performant on other, and connect to your workflow of choice, right? That such additional flexibility makes a lot of sense.
Okay, thank you.
Okay, are there any other questions?
I just have a general question about future industrial collaborations, and if you think you could, you know, as at EMBL or anywhere, if you could push or try to gamble different vendors for more broad availability of API, or is this a really hard challenge? Okay, can you go to Zeiss and say, you know, we can't get this availability, we'll switch to Nikon, or is it what would it take for companies to really open up more? Do you have any insight into this? Or how big do you have to be to push companies?
Yeah, I think it's a fair question, but it's difficult to say because, of course, you don't want to argue with a company by naming another company. So again, I refer to the working group, which is currently on the slide, because we have like a number of people from facilities, but we have also company representatives. So we have Zeiss representatives there. We have, I mean, certain point Leica representatives were also joining. We have also the representatives of smaller companies. So the idea is, by having this working group, to speak not on behalf of one facility, but speak on behalf of more than one facility, or hopefully the community, right, at some point, to make a point that it's not only for EMBL, right? It's basically important for the entire community. So if you're interested, so the participation is totally open. I mean, it started here, right? But maybe there are parallel initiatives exist on your side, right? Or maybe if you're interested to join or participate in this meeting, so you're welcome to contact us via email, right? Or contact the working group via email, right? And have a look what's going on there. So more we act as a community, probably more we push in this direction. And then basically we make a point that is important on different sides.
And this is so cool and so important. Great.